In today's video, we're going to be talking about how to analyze bioinformatics and the information that they provide. So, as you know, we have done DNA sampling in the class, and all of that will go into this. So, all of that DNA is being used to create this data in this chart. So as you can see, we're going to be analyzing first substitution rates, which you can see on the screen here. So we have to see which strand is best for substitution. So this is all kind of based on how they evolve based on evolution. So as you can see, the shape of the parameter of the gamma distribution means that a small parameter could suggest that certain regions of that sequence are under more selective pressure or constrained, that is. So our evolutionary results show us that we have a large parameter, as you can see on that graph in the bottom left-hand corner. And this is telling us that most of these strands have evolved at similar rates. Here is the maximum parsimony tree, and this kind of shows that all of these bacteria that we got from our agar plates are all related in some way, so they all share similar sequences. So my bacteria personally was Pseudomonas aeruginosa. So this is related to all of these other bacteria that are in this tree. So this tree is just kind of showing how they are similar based on their evolution. And this is our neighbor joining tree. So as you can see, a very similar build to the MP tree, but they both show a little bit different things. So they show us similar relationships in each of these trees, resulting in similar outcomes. So it's kind of showing that evolutionary background. But the difference between the two is that the MP tree is more focused on providing the most likely evolutionary pathway, as we saw on the last slide, while this tree and this data is more for inferring relationships based on genetic distances. As you can see there at the bottom, we see our distances, and the fourth one from the bottom is actually probably the best one because it's closest to 0.5. The next step that we did in our bioinformatics was test for Tajima's test of neutrality. So what this means and what it's showing us in our diagram is that many of these strands were close to 0, which means that our sample was close to having natural evolution. As you can see, the frequencies are varying between the 1, 2, and 3, and that just means that we are seeing more of that first strand than our second or third. But as you can see, Tajima's D there under primary metrics is negative 0.73. That is very close to 0, showing that we were close to natural evolution, but because it's negative, this could mean mutations were removed during the purification of the DNA sample. One of our final steps in bioinformatics was testing the molecular clock. So as we are following this data set, it says that the parameters are just over 0.05 CVs and under 1.0 CVs. So this would suggest clock-like evolution. And this matters because deviation from that can find us in different events and different mutations that can happen during the evolution process. So as you can see, our standard deviation rate is 0.053. This is just over the 0.05 mark, which means that the DNA that we collected was, in fact, clock-like evolution because it doesn't deviate from the set numbers, as you can see here on all the graphs and also on the interpretation guidelines above.